GSK. We're going to go ahead right into the FDA presentation. So first will be Dr. John Alexander.
Good afternoon. I'm here to discuss the breakthrough therapy designation for Tafenoquine. So Tafenoquine is an 8-aminoquinoline antimalarial drug. A lot of the antimalarial drugs that are available are quinoline derivatives. This one is a synthetic analog of primaquine, which we'll talk a little bit about later in the presentation. It has activity against all stages of the Plasmodium vivax life cycle, including the dormant stage, the liver stage, the hypnozoite, and that becomes important in discussing the results later. Because of the drug has slow clearance of the blood stage, it therefore requires co-administration with another faster acting blood schizonticide, such as chloroquine. So a little bit about the disease, Plasmodium vivax malaria. Vivax malaria is considered a serious disease, although it may not be as fulminant as other forms like falciparum malaria. It still is considered a life-threatening disease if untreated. There are 130 to 390 million cases annually worldwide, and it accounts for approximately half of the cases of malaria worldwide. P. vivax has a complex life cycle, which includes this dormant liver stage, the hypnozoite stage. And this is important in the disease and the treatment that we're considering because it's reactivation of this hypnozoite that leads to relapses of vivax malaria in particular. So in terms of treatment of Plasmodium vivax malaria, the treatment involves two drugs. The treatments that are approved now involve two drugs, the use of chloroquine followed by the use of primaquine, usually in doses of either 15 to 30 milligrams daily for 14 days. And it offers what's called radical cure, which in essence means not only do you eliminate the disease that's present, but the radical cure is the prevention of later relapses. So in terms of the drug that's being used as a comparator in this presentation, FDA approved primaquine, a 15 milligram daily dose back in 1952. And it's the only other available therapy. So for Tafenoquine, the sponsor requested breakthrough designation in October of 2013 during phase 2 development. The indication, as we discussed, was treatment and relapse prevention, radical cure of Plasmodium vivax malaria. We had an end of phase 2 meeting with the sponsor in November of 2013 where we discussed the phase 2 clinical trial results and the proposed phase 3 clinical development program. The breakthrough therapy designation was granted on December of 2013, and there was a medical council meeting was held to discuss the Tafenoquine application. So the clinical trial results from the phase 2 study were the main results that supported the breakthrough therapy designation. There were results from 3 phase 2 trials altogether that were submitted to support the sponsor's request, though. As I said, the main results come from the phase 2B trial, a multi-center, double-blind, randomized, active control dose-ranging study to evaluate the efficacy and safety of chloroquine plus Tafenoquine in various doses versus chloroquine alone, and also with an active comparator of chloroquine with primaquine given at the 15 milligram daily dose. The study was conducted in 329 adults with malaria. All of the patients in the trial received the same dose of chloroquine. The negative control is the arm with the chloroquine alone. And there were varying doses from 50 to 600 milligrams of Tafenoquine given as a single dose after completion of the chloroquine treatment. So these are the results of the phase 2 study. The column all the way on the left is the results for the chloroquine alone arm, and these are results that are showing the relapse rate I'm sorry, the number of individuals who are relapse-free after 6 months. So in the chloroquine arm, the number who were relapse-free was around 37%, indicating that roughly 63% of the patients actually relapsed during that 6-month period of time. The 4 columns in the middle are the varying doses, 50, 100, 300, and 600 milligrams of tafenoquine given as a single dose after chloroquine treatment. And then the column on the right is the column showing the treatment with chloroquine plus primaquine. The p-values that are provided are all comparisons to the column on the left, the CQ alone arm. And what you see is clearly a dose response where you have better response at the higher doses in terms of fewer patients who relapse with either the 300 milligram or 600 milligram dose in comparison to the chloroquine alone or the lower dose arms. The other factor to note with this, though, is that there was 77% of patients were relapse-free in the primaquine arm versus 89.2% in the 300 milligram dose, which is going forward into phase 3. So we did see a 12% better rate of remaining relapse-free. So there were 2 additional clinical trials that were mentioned where we had results. There was an open-label dose-ranging study, 7 dose regimens of tafenoquine ranging from 500 milligrams to 3,000 milligrams, so much higher than what's proposed to be used in the final trial. Again, compared to chloroquine plus primaquine and chloroquine alone. And again, in this trial, the varying doses of tafenoquine, they also received chloroquine treatment. So the relapse rate for the patients in the chloroquine plus tafenoquine arms ranged from 0 to 13%. The relapse across treatment arms for the chloroquine plus tafenoquine when you add them together was about 4%. For chloroquine alone, the relapse rate was 71%. For chloroquine plus primaquine, the relapse rate was 25%. So again, suggesting that there was an added benefit of the chloroquine, of the tafenoquine treatment arm versus the standard treatment. The other study that was available was a randomized active controlled double-blind trial of tafenoquine here given as monotherapy, given 400 milligrams per day for 3 days, in comparison to, again, the standard treatment. The tafenoquine monotherapy in this study showed us a slow effect on the blood stage, which is why it's proposed to be used with chloroquine in the other trials that were conducted. But the tafenoquine had a high rate of remaining relapse-free up to 4 months after the treatment. So in this case, the rationale for the breakthrough therapy designation, Vivax malaria is serious disease. The preliminary clinical evidence demonstrates that tafenoquine provides what appears to be a substantial improvement over the primaquine treatment. The tafenoquine provides an alternative treatment option with a shorter dosing regimen, which may be valuable in terms of directly observed therapy in areas where malaria is endemic. And the single dose therapy should be able to also improve compliance compared to a 7 to 14-day course of primaquine. Thank you.
Great. Next, Lucas Kempf.
Hi, I'll be talking about the case study of Nobin for the treatment of negative symptoms for schizophrenia. So just a quick overview of what I'll be talking about. So schizophrenia is obviously a serious condition. It affects about 1% of the world population. It's a chronic disorder that starts in late teens, early 20s, and is highly debilitating to the individual. It is characterized by, it's considered a syndrome, so it's characterized by chronic positive symptoms, meaning symptoms that come de novo to the patient, sort of in a positive sense, not as positive as it's good to have these. With the hallucinations, delusions, and thought disorder, and the negative symptoms, which are things that are taken away from the person. A flattening of the affect, anhedonia, amotivation, ambivalence. And also along with it, typically when a patient has their first episode, they have about a 10-point general IQ loss and develop problems with frontal lobe processing that involve planning, memory, processing speeds, and other cognitive domains. So currently, for the last 50 years, primary drug development in the realm of schizophrenia focused on dopamine receptors and dopamine blockade. And what we've seen with these therapies is they're primarily just improve the positive symptoms with very little improvement in the negative and cognitive symptoms, leaving the patient with this sort of residual deficits in cognition and negative symptoms. So there's currently nothing on the market that targets these. So the sponsor came in asking for breakthrough designation for negative symptoms based on a single study. Now, this study was published in JAMA Psychiatry recently. It was a randomized, double-blind, placebo-controlled trial of 52 patients with chronic schizophrenia who had been stable with their symptoms on a stable dose of at least 3 months of a typical antipsychotic, and with a minimal PANSS score of 60. So the PANSS score is a measure of both positive and negative symptoms and general pathology of the illness. So this was a 6-week add-on trial to the regular therapies of 1 gram of sodium benzoate or placebo. Their primary outcome measures was the PANSS, which is our typical measure that we use for any antipsychotic that's come on the market. Additional outcome measures within the study were cognition, scales to assess negative symptoms, global assessment of functions, quality of life symptom scales, depression rating scales, clinical global assessment, and the Matrix Consensus Cognitive Battery, which is a cognitive battery that was developed to help drug companies target development for these cognitive symptoms. That was in development with the FDA, the NIH, academia, and industry. It was a series of meetings that took place over a year to kind of come to a consensus of what we would actually accept for improvement in the cognitive space. So here are the results from the study as published. So you can see that sodium benzoate improved the total PANSS score, highly significant p-value, improved just breaking it out into the subscales, the positive symptom scales, the negative symptom scales, and the general psychopathology scale. And then further on, since we've had no drugs that actually have improved negative symptoms, if you look at the SANS, another measure of negative symptoms, they had a highly significant effect on that scale. Global assessment of functioning, quality of life, CGI, and the depression scale, all of which were pointing in the same direction and highly significant. Then when they moved to the measurement of the matrix, which was this battery that we helped design, and nobody's been able to win on this, they won on some elements of it, which you can sort of see better here, where they won on the processing speed and the visual learning memory subcomponents of this test. And then if you average that all together, they also won on the composite score, the neurocognitive positive composite score, which takes out the social cognition score, which there's been a lot of debate in the literature whether or not that scale that's incorporated in the matrix is actually going to be sensitive even if somebody develops a drug that targets that. So within the division, we had a discussion about this claim for negative symptoms. So previously, we've outlined a pathway for negative symptom in schizophrenia indication for approval back in 2005. And as part of this discussion, if somebody was going to win for a negative symptom indication, we wanted to make sure that that was just specific for negative symptoms. That if you ended up creating a medication that improved everything, that would be considered a broad spectrum treatment. And under the current state, we don't have anything that broadly improves schizophrenia. So our rationale for this breakthrough therapy was that what we actually had here was a medication that improved the positive symptoms, the negative symptoms, and the cognitive symptoms. So we actually denied the narrow claim for negative symptom indication. And then we had a meeting with the sponsor and explained this rationale, and they amended their application to ask for the adjunctive broad treatment of schizophrenia. And then we also went back and looked at previous antipsychotic trials to see if you just improved the positive symptoms in those studies, what effect you would have on the other aspects of the negative symptoms. And the effect size that we saw with NABEN was larger than what we saw for the effect size if you just improved positive symptoms. That meaning, so if you have a patient who's highly hallucinated, they're predictably going to do poorly on any sort of other test. And if you improve their hallucinations or delusions or their disorganized thought, they might actually be able to be evaluated better. So you always see a slight improvement on negative symptoms or cognition as you improve the positive symptoms. And then in addition to this, there was no SAEs in any of this program. Sodium benzoate is generally recognized as safe molecule. You can take several grams of this in your daily diet without much problems, and the side effect profile within this was fairly benign.
Great, thank you. Next we have Dr. Eileen Irani to present the case study for Lentiglobin BB305 from Bluebird Bio.
Thank you, and good afternoon. So I'm bringing a case here, a case example for CBER, for Center for Biologics, on a gene therapy. And the slides here are a combination of my work and our work and the sponsor, Bluebird Bio. And so I'm going to talk about the disease overview, what's beta thalassemia major, which is the point that we will be discussing for this case example. The specific product, the description of the gene therapy that's proposed for treatment, a summary of the clinical evidence that was necessary for the breakthrough designation and for the decision. A little bit I'm going to talk about the sponsor's lessons learned, but I'm going to leave most of this for the discussion with Bluebird Bio afterwards and our FDA considerations. So first about the disease. So we're talking about a rare disease in the US. Beta thalassemia major is transfusion dependent, is a monogenic disease that results in severe anemia. Patients have multiple mutations in the gene for the beta globin protein, resulting in either absence or significant reduction in the protein. And that results in an imbalance between alpha globin and beta globin in the red blood cell and causes instability of the red blood cell, hemolysis, and therefore substantial anemia and consequences of anemia. Plus some, because of the hemolysis and myeloid, bone marrow proliferation and extramedullary erythropoiesis, there are some additional problems. And there's a constellation of other symptoms and problems that diminish both quality of life and lifespan. And it's a significant problem. What the global prevalence is about 280 to 190,000 patients with this form, with the severe form of beta thalassemia, transfusion dependent. And in the U.S., this was granted orphan product designation a few years ago based on a calculated or estimated prevalence of 5,600 patients in the U.S. as a prevalence. It affects mostly people in the Mediterranean areas, as well as Southeast Asia and South Asia, Pakistan and India in particular. The standard of care for transfusion-dependent beta thalassemia major is the constant need for transfusions, blood transfusions. Without them, people develop severe anemia and all the consequences, including death. With the frequent blood transfusions, there is a need for prevention and treatment of iron overload with iron chelation therapies, which are sometimes difficult to tolerate, and there are some issues with compliance, long-term compliance with the iron chelation therapies. And that can lead to iron accumulation and the pathogenesis related to iron accumulation. The other potential treatment is allogeneic transplantation, and that's, it's rarely used primarily because of the risk-benefit of this option. It's mainly used for patients that have a fully matched HLA, fully matched sibling donor, and still, despite this, there is some chronic morbidity, graft versus host disease, rejection of the graft, and potential issues with chronic immunosuppression from the bone marrow transplant. So there is clearly, it's a serious life-threatening disease with with an unmet medical need. The product itself. So this is a gene therapy. So Lentiglobin BB305 consists of autologous CD34 hematopoietic stem cells that are transduced with the lentiviral vector that carries this beta-globin gene. The beta-globin gene here that was studied, it has a particular marker. It's a one-codon mutation that doesn't affect the function of the beta-globin protein, but serves as a marker for us to track the expression and the effect of the treatment. So it's a substitution of one amino acid in the beta-globin gene. It's been developed for both beta thalassemia major as well as sickle cell disease, which is another condition that affects the beta-globin gene with a very specific point mutation, but with a different constellation of very serious problems. But the presentation now will focus only on the breakthrough designation request focuses solely on the beta thalassemia. So this is just a figure at the bottom showing the vector itself with the human beta-globin gene in the middle, and the in vivo biomarker is this electrophoresis showing a peak of the beta-globin gene on the graphic on the right. So I'm going to talk a little bit about how this is made. So first you get the plasmid that you see on the top of the step 1, and this is introduced into a packaging cell line, human cell line, and the result of this first step is the lentivirus itself carrying the plasmid of interest, a transgene that's going to be substituting for the defective gene. The second step is the autologous CD34 hematopoietic stem cell procurement. So for this, patients are treated with filgrastim or plerixafor to mobilize hematopoietic stem cells to the peripheral blood. There is an apheresis process that collects CD34 positive stem cells and retains those cells. Step 3, now it's transduction of those hematopoietic stem cells, CD34 positive, with the lentiviral vector that was produced in step 1. So that takes around 48 hours. And then you see some modifications in those stem cells, the hematopoietic stem cells that start to express the beta-globin gene. And the last step is the actual treatment of the patient. So reinfusing back those CD34 cells to the patient after full myeloablation with busulfan. So there's a treatment with busulfan for doses in 4 different days, and transfusion of those CD34 cells, and then we wait for engraftment and for the clinical effect of those modified CD34 cells. So, as I mentioned, there were 2 requests for designation, for breakthrough designation. The initial breakthrough designation came right after FADESIA was approved, or a few months after FADESIA was signed into law in PDUFA 5. And Bluebird Bio came with clinical evidence of 3 patients that were treated with the previous product, a different vector, which is Lentiglobin HPV 569, and some non-clinical comparison between the lentiviral vectors that was mainly in a murine model of beta thalassemia. And from those 3 patients, the evidence was that 1 became transfusion independent by 1 year post-transplant, and that was sustained for 5 years. 1 patient did not engraft and received the backup cells. I forgot to mention that when cells are transduced, there's a backup pool of hematopoietic stem cells that are used just in case there is a problem with the transduced cells. And so the second patient received the backup cells to engraft. And the third patient remained transfusion dependent, so the treatment didn't work for that. So, on the bottom here, you see a comparison between the 2 vectors. The main difference between the previous vector and the current vector that Bluebird Bio is using is there were small modifications, but the main one was the deletion of 2 insulators that were thought to be interfering with effectiveness of the transduction of cells. So the lentiviral lentiglobin was not being effectively taken by the cells, and it wasn't an effective and consistent expression of the gene protein. So at the time of the denial, we explained that in the absence of any clinical data with the current product, the BB305 product, the requirement for the preliminary clinical evidence is not fulfilled. However, we said the data were promising. And here there was some discussion about, well, there were 3 patients treated, and 1 patient was transfusion independent by at 1 year, started at 1 year, and then that was sustained for 5 years. How much of that evidence was sufficient or not in a rare disease to justify breakthrough therapy request? But the issue was moot because of the change in the vector. And here the clinical evidence when the reapplication came, with the new request for designation came. So Bluebird had conducted 1 study in France, single center in France, the study 205 with 2 patients, and 1 study involved the US, Thailand, and Australia. And that second study, 204, is under an IND. And you can see here the demographic characteristics of those patients. Most of them are, or all of them are between 16 and 26. And the majority of the patients are female with varying phenotypes, but most of them with varying genotypes, but most of them, or all of them indicating severe genotypes that are leading to transfusion requirements that are very high in the diagnosis of thalassemia major. And the transfusion requirements here are, as you see, for inclusion, they have to be greater than 100 mLs per kilo per year, which translates in approximately 8 to 10 transfusions a year at least. So every, or even more than that. I think between 3 weeks on average, you need to have a blood transfusion. as a requirement for eligibility. And here you can see the results in 4 patients that completed at least 3 months of, after the treatment with gene therapy. So 2 of the patients are in the US study, and 2 of the patients are from the French study. And you see here, what's in blue is the hemoglobin A, which is a normal adult hemoglobin as a result of the last few transfusions or the last transfusion that the patients received. And gradually that hemoglobin A decreases over time, as you would expect for someone who is not receiving blood transfusions and who doesn't have the normal hemoglobin, globin gene. On the other hand, you see that gradually the fraction that's red on top is increasing, and that fraction of red is the expression of the beta globin gene with the marker, with the AT87Q, the threonine glutamine substitution. And you see that this is increasing and maintaining the overall levels of hemoglobin above a threshold level of 9, which is a commonly identified threshold for transfusions. And you see this, there's a consistent gradual increase in the expression and stability of that marked tracked hemoglobin AT87Q, which is an expression of the gene therapy. This is another representation here showing the month post-drug product infusion, showing that these 4 patients are transfusion-free, regardless of their genotype being beta 0, beta E, beta 0, beta 0, for example. All of those are for varying durations after their infusion of the gene therapy. They became transfusion-free. And at the time of the review of the second request for breakthrough designation, we had asked for more current data and additional data. And Bluebird Bio provided this, showing that there was no new surprises, no negative surprises in terms of what they had demonstrated before. And for the 4 patients that had more than 3 months of treatment, they were all transfusion independent after treatment. And they gave us also the graphic representation showing that before treatment, what their blood transfusion requirements were, and after treatment, which are 2 of the patients are listed here in the bottom graphs. So as far as our considerations, the main issue here that we're going to bring is that for gene therapies, because the treatments are so novel, it's very hard for us to understand small modifications sometimes in the vector, how consistent, how similar the products are. And for the breakthrough designation request to be granted, it has to be with the product that the evidence has to be with the product that is being studied and is being developed, not a previous product. And we were not sure by removing those 2 small insulators if the product was substantially different or not, both in terms of efficacy and in terms of safety, in terms of risk of insertional mutagenesis and oncogenicity, etc. So that was one of the questions that were raised between the first request for designation and the second one. And the other issue is, I think that was alluded to in the context of rare diseases, is not about the statistics, it's about the magnitude of an effect or the treatment effect size. And here, there was a clinically relevant endpoint, so there was no biomarker here. This is a transfusion independence is a clear clinical benefit, and this was met even in a few patients that were already treated. So in terms of safety, I just want to say a word of safety. There were no surprises here. Busulfan had some toxicities on this conditioning regimen for myeloablation that is necessary for engraftment of these transformed CD34 cells, and we saw those busulfan toxicities less than grade 3 NCI common terminology criteria, but nothing else.
Great, thanks, thanks Elon, and thanks to all three presentations. Those are very, very helpful. I think what I'll do, I think what I'll do here, since we have three very different therapeutic areas, is first turn to the, in order, the manufacturer representatives from the companies that submitted those products for breakthrough designation, and then I'll quickly ask the rest of the panelists if you have any comments based on that particular case, and then I'll go to the next case and the next case, and then I'll take it up a level if we can think of some themes and lessons learned across the therapeutic areas. So first I'll turn to Andy Gustafson from GSK, and we saw in the presentation that the Tafenoquine demonstrated, significant improvement over chloroquine, but then we also saw only a 12% improvement over primaquine. So just thinking of that, and like to better understand what you all were thinking when you decided to go after the breakthrough designation. What were you how were you weighing the evidence, and then maybe comment on that progress.
Okay, sure. Thanks, Greg. Make a few comments on Tafenoquine, which we're developing along with Medicines for Malaria venture. And as you probably know, P. vivox malaria in the U.S. is a rare disorder. But as John showed us, outside of the U.S., in areas like Eastern Asia, Africa, South America, there are hundreds of millions of patients each year that are affected by the disease. So in terms of the timing for our breakthrough request, similar to some of the other sponsors that you've heard from today, we had already been in discussions with the FDA early on in the program. And in fact, we had our end of phase 2 meeting in the month before the breakthrough was granted. So when we received our letter granting the designation, there's a statement in there advising the sponsor to have a Type B meeting and to come in quickly. Well, we didn't need to do that, since throughout the program and even the month before, we had very detailed discussions about the phase 3 program. So as you've heard from others, our motivation for applying for and obtaining the breakthrough designation was really to benefit from the intensive communications and the more senior management involvement as we went forward from phase 3 to the time of submission. In terms of the evidence for the breakthrough designation, I think John covered that thoroughly. It was very important, as you mentioned, for us to include in our phase 2 trial that arm with the existing therapy, the primaquine, along with the chloroquine. So we had a benchmark where we could compare and demonstrate the potential for improved efficacy. And then we also have this Tafenoquine with its extended half-life, which is on the order of 15 to 19 days, which allows for a very unique one-time dosing regimen and allows for the potential for increased compliance, which we hope would translate into improved patient outcomes. And when considering the geographic regions where the medicine would be used, we think is a very important feature to the medicine in terms of its potential. So in terms of our experience with the breakthrough designation since, our experiences working with the FDA since the designation, I think we feel our interactions have been very constructive. We've been able to talk with the agency about streamlining our program in areas outside of the phase 3 program, particularly in terms of other clinical studies, special studies that we're going to do leading up to the submission, and also in the CMC area in terms of the manufacturing area. So overall, we feel the process has been very helpful and positive, and will help us to maintain alignment with the FDA as we go forward.
Thanks, Andy. That seems to be a unique consideration of the primaquine being once a day for 14 days, and the Tafenoquine being just a once a day or one-time dose. That the context of that is in the countries that the last point that you just made is that becomes an increasingly important consideration in terms of adherence and compliance. I'd like to just ask the rest of the panelists any particular questions or comments on that issue or that product. John Powers.
So I did want to bring it into that context of what do we mean, and we've been talking about this most of the day, by substantial improvement. And to me, I think there's three categories that really apply to FDA and one that doesn't, but I'm going to bring it up anyway because it's something for sponsors to think of. The first one, and then we saw the data on Tafenoquine, was improved effectiveness. And interestingly to me, almost the entire discussions through all of the examples today have been on examples of improved effectiveness. The second one, though, is decreased harms. And it strikes me that perhaps presenting preliminary clinical information on decreased, or on an agent whose primary substantial improvement is decreased harms might actually be quite different. And the reason for that is that if your benefit is improved effectiveness, people might be willing to accept actually increased harms in that situation, much like oncology drugs. Whereas if your benefit was decreased harms, you'd want to try to preserve some of the benefit of the older agent as well. But it strikes me that we haven't discussed much what would preliminary clinical evidence look like if the benefit was decreased adverse effects. The third one is something we just talked about here, and that is when does improved convenience become an actual substantial improvement? And we picked a situation which is probably unique in that we know something about malaria. For instance, there's a randomized trial that actually shows that 5 days of Primaquine was similar to placebo. So if you take it for less than the prescribed 14 days, there is a clear decrease in effectiveness. We don't know that for the vast majority of infectious diseases. In fact, for shorter-term things like pneumonia, there are studies which show that 5 days may be similar in effectiveness to 10 or 14 days. So if you came in with a new agent whose primary goal was to shorten therapy for pneumonia, what does that mean? So the point I'm making is, should improved convenience actually result in either increased effectiveness or decreased harms? So in other words, a drug that just goes from twice a day to once a day without changing either of those other parameters, is that really a substantial improvement? The last one I touch upon is cost, which is not an FDA issue. But in some of the things, Greg, you've held on antibiotics recently, we've discussed this issue of payers wanting to know that the improved effectiveness or decreased harm translates perhaps into decreased cost. And FDAMA Section 114 actually allows sponsors to present that kind of information to the appropriate audiences. So it's something to think about while you're gathering the information, to try to think about what the payers are going to see beyond what the regulators might want to actually see. The last thing I wanted to bring up was this issue of clinically significant outcomes. And again, that might be very different in an acute disease like pneumonia versus a more chronic disease like malaria. And that is that we need to think about, and I think Ellen Siegel brought this up earlier, people with most types of infections, and malaria is not one, unfortunately it doesn't fit this example, are highly symptomatic. So say people with chronic lung infections. And could we build in information on patient-centered outcomes, like patient-reported outcomes, into those in addition to things like biomarkers? A lot of discussions about biomarkers today. But in fact, the definition of an invalid biomarker is one in which it shows a positive effect, but there's actually no effect or a negative effect on how people feel, function, or survive. So depending upon, and that leads me to my last thing, this is all contextually dependent, right? So the things that apply in malaria, like this application, may not apply in a short-term acute disease like pneumonia, and you have to think about those things differently. And finally, I asked the question this morning about 72% of the applications that were turned down were because of trial design issues. And it seems like there's a legitimate response to that many of these applications were actually underway before FDASIA was actually passed. It would be interesting to go back 2 years from now and look and see whether guidance from FDA actually impacts that. Because I have to say, having worked at FDA myself, I found there were two categories of sponsors: those who took FDA's advice and those who did not. And so sometimes what happens is, I always thought it was ironic to listen to at advisory committees where a sponsor would get up and say, we discussed this with FDA. Yeah, my kids discuss doing their homework with me too, and they don't always do it. So I think the fact that you discussed it doesn't mean you followed that advice. And the reason why this is even more important is that what's in the guidances on FDA's website may not really apply to game changer interventions. You may have to do things differently than the standard way of doing things.
Okay, thanks, John. Any comments to that or to this particular case? Okay.
I guess
go ahead,
just
John.
add one comment with regards to the idea of how this is going to affect clinical trials in the future, because one of the things that I think is there might be a positive effect of the potential for breakthrough designation if you understand enough of your drug in the first place when you're in a preclinical phase or an early phase 1, that there's the potential for your drug to be a game changer. It might actually promote better work on phase 2 studies. In the anti-infectives division where I work, a lot of the products go almost directly from phase 1 into phase 3 because a lot of the modeling that's used to sort of target what dose they think will be effective can be done without the need for sort of a phase 2 trial to promote that. And this may be the case where if you're really thinking that your drug is going to be a game changer, the idea of spending time exploring what goes on with your drug in phase 2 could actually be something that will translate into a benefit in your being able to get more interaction with FDA and get a earlier timeline for getting your drug through the approval process.
Okay, great, thanks for that point. I'm going to turn to Emil Tsai. Your company had originally applied, what was interesting in looking at the results of NABIN is that ultimately the clinical data that were supplied showed substantial improvement on negative symptoms, positive symptoms, and cognitive symptoms, but yet the original indication for which you applied for the breakthrough designation was just the negative symptoms. So I was wondering if you could walk us through sort of that thought process and what led you to first seek out that more narrower indication.
Sure, I think I can explain my background. So when I talk about my thinking process, that can answer the questions. Myself is a UCLA professor in psychiatry. I'm also the chief scientist of this company called Cinerex. My training is in psychiatry and in neuroscience. So when we get the data of Dr. Kemp presented today, of course we are very happy because we know we meet the criteria, one of the criteria called substantial improvement. That's very clear. But it's not clear to us is that whether we have meet another criteria called serious illness. The reason is that, for example, when I consult a regulatory colleague, I say, maybe we should try this. And the answer I get is, that's not for psychiatric drugs. It's for oncology drug because mental illness, they don't kill people. Mental illness just, people live a lousy life. There's no mortality come with that. But immediately my academic background kick in. I say, this is not the case because I know, 10% of schizophrenia, they commit suicide, We know there's a lot of illness are refractory, including depression, including OCD, dementia earlier, and also even ADHD, you name it. It's a long list for mental illness. So at the time we think, but I'm still in doubt. This person suggesting that it's only lethal illness can get a breakthrough. So I'm thinking maybe the FDA will ask me whether I have a suicide outcome measure, which will be impossible to show. So at the time, I'm from the academia, so I know where the consensus is in academia is that what the next treatment need to be. And in the past 20 years, we've been searching for kind of holy grail treatment in schizophrenia. And we identify negative symptom, cognitive symptoms as our next generation treatment goals. And also Dr. Kemp also lay out one of the examples is a joint effort by FDA, National Institute of Mental Health, or even EMA, European Medical Agency. They had come together 3 times in the past decade to develop a kind of consensus of how to develop treatment for negative symptoms. To make that long story short is that we go through all the consensus and we think we are consistent with it. Only one point is that there's a hypothetical notion there suggesting that new treatment good for negative symptoms should be for negative symptoms only. I look at that, I'm not sure, our candidate can fulfill that criteria because I understand it's a hypothetical you want your treatment only for A, not for B, but it's a syndrome. So it's hard to tease it out. When it happens, it's good for both. So at the time, we say, maybe I'll just give it a trial and for negative symptoms, because we do have a very good signal about the negative symptoms. And we know that a lot of company has done use the same mechanism of action to develop new drug, but most of the trial are fail. It's not, they turn out to be negative because a lot of trial in psychiatry product they fail. They don't, it's not they are not effective. It's the most notorious is a placebo response. So we realize this is a game changer, and we realize we got substantial improvement. We're not sure about, serious illness, but we think negative symptoms should meet that. So that's why we applied for negative symptoms. But it's a very nice surprise. We come back with a very good comment and saying that, this is add-on treatments. And it's also a bias we have at the time, because I'm a practicing psychiatrist. In real life, we do add-on treatment all the time, off-label. We do that. Most of the schizophrenic patients, they are on polypharmacy, we call it. They're not on one antipsychotic, maybe on 3 antipsychotic. So add-on treatment is not novel to practicing psychiatrists, but for regulatory field, it's novel because there's nothing being approved for add-on treatments. So we were very friendly reminded by FDA that this add-on treatment is also going to be important treatments for the populations. So at the brief conference we have with the FDA, we are very happy to accept this revision of the indication from negative symptoms to add-on treatments for overall symptom profiles. So for the future, we kind of want to look forward to use this as a good opportunity to work with FDA to look for a better enrichment approach that can minimize the failed trial rate, in psychiatric drug products. That's my response to your questions. Thank you.
Okay, great, thanks. Any other comments from the rest of the panel on that case in particular? Okay, so I'll turn to AV. So your company, similar to Area Pharmaceuticals this morning, experienced an initial denial of the request and then subsequently were granted, and that provides a unique opportunity to sort of hear from you and to learn what you all learned from that process and experience in going through those particular applications.
Sure. So before I go through our lessons learned, I had a couple points. To me, the discussion so far have highlighted the increased need for dialogue with the agency, and I think we welcome this workshop because I think it will be very helpful for future sponsors to file successful applications. And in many ways, the past few years since FDASIA has been enacted can be seen as a pilot, and I think it's critical to learn. And this is why Bluebird volunteered to share our experience. We encourage the FDA to perhaps put a link on their website to the workshop or to a summary. I think that would be helpful for sponsors, in particular small companies. Our lessons learned are multiple-fold. First, the Office of Cellular Tissue and Gene Therapy, OCTGT, has been very helpful throughout this process, and we learned a lot already from the denial of the first application. As Dr. Irani explained, we learned that the clinical data really had to be with our product and our manufacturing process. So the FDA did not consider the data coming from the French trial because that was not under the IND. So that was helpful. But most importantly, in the denial itself, there was some recommendations and some proactive comments from the FDA, particularly with regard to the endpoint, for example. And the OCTGT clearly said that the endpoint we had selected of transfusion independence was the right endpoint. So that was helpful for us for the future application, but also in the way of thinking of designing our trials. So that was actually very helpful. And that really opened the door for further dialogue with the agency for our next application with regard to the number of patients that we would need and the magnitude of effect. So for us, it was really a positive experience. And similar to GSK, our next interactions with the agency after receiving breakthroughs have been very positive as well. And we have seen the advantage of having breakthrough with regard to the interest and the attention that was put into the review of our documents and the feedback that we received in terms of how can we accelerate the development. And I have one final comment. This morning, I think someone asked whether the bar was too low. I think from today, I would say that from my perspective, it's not. I think the designations that have been granted and the case studies that have been presented have been very helpful and well thought through and justified. I think the FDA could consider doing further things for products that have been designated with breakthrough designation and thinking about how can they have further discussions on biomarkers that could be helpful, or perhaps thinking of adaptive pathways, which are things that are considered in Europe, and that may be also helpful for accelerating further the development. Thank you.
Okay, thanks for that. Any other comments or questions from the panelists on that particular topic? I'll turn to David Fagenbaum. So David, you're a medical researcher at Pennsylvania. You're also a patient, and I'm wondering your perspective on all of this. I know you weren't able to hear the earlier panels, but particularly for these products, any reflections from you on the breakthrough designation process and the way that the evidence appears to be weighed?
Absolutely. Thank you, Greg. So just as Greg said, well, first off, thank you for being a part of this. I guess I say that on behalf of the patient perspective. Thank you for the time that you all put into advancing drug development. So just as Greg said, I'm a research professor at UPenn. I lead a research network called the Castleman Disease Collaborative Network. I've actually served on an advisory board for a pharma company developing a drug for Castleman disease, and then I'm also a patient with idiopathic multicentric Castleman disease. So I guess I wear a few different hats in this game. So most of my personal research is actually focused on identifying and discovering biomarkers for multicentric Castleman disease and also establishing a clear natural history for the disease. And so I think I was able to be a part of the neurology panel just before this and hearing this one. It's just become abundantly clear how important it is to have these validated biomarkers and certainly clear understanding of natural history, which is a real challenge for orphan diseases and something that I certainly saw when I first got introduced to multicentric Castleman disease as a medical student when I first became ill. But when I came back to medical school, it became clear, and today's meeting is highlighting that, just how much work needs to be done in the orphan disease space to establish and validate biomarkers, to clearly define natural history. And I'll go through a couple pieces or a couple steps that we've gone through. So now I'm talking on behalf of the Castleman Disease Collaborative Network, a global research initiative that I founded a few years back. So really with no natural history study, no validated patient-reported outcomes, poor understanding of how the disease works, no clear subclassification system, pretty big mess. We started out by saying instead of funding research with this really broken system, let's actually start by focusing on the system of biomedical research for Castleman disease. And so we began by identifying and uniting the global community of physicians and researchers doing work for multicentric Castleman disease, and then using that community to identify and prioritize what research should be done next. So really, rather than taking the old model of raising as much money as you can as an advocacy group and letting researchers come to us, we said, let's actually go to the researchers, let them prioritize what should be done next, and then target, in a targeted fashion, strategically, do outreach to individuals around the world who are conducting that kind of research. And so I think that this kind of targeted approach, we're on our way to identifying biomarkers. We have 3 studies right now that we've launched. And I think that this targeted approach is one, going to help identify these biomarkers for future therapeutic development, but it also builds the infrastructure that we'll need to do drug development in the fashion that you guys have all shared. So I think that maybe I want to conclude by, again, on behalf of patients, thanking you all for what you're doing to develop drugs for patients like myself. I think I also want to just highlight that from the patient perspective, in the words of Dr. Janssen, where Janssen gets their name from, patients are waiting. And I guess also in the words of Maria from the Alzheimer's Foundation, we'll take anything we can get. And so I hope that you guys will continue to do the work that you're doing, because a lot of us are waiting, and we hope for progress.
Great, thanks, David. I'll turn the next question to John Jenkins down at the end. John, you're part of the Medical Policy Council representative. The three cases presented here bring a lot of different issues. The Tafenoquine has an interesting endpoint of transfusion independence. NABIN, also in terms of endpoints, used functional and quality of life endpoints. The gene therapy, Lentiglobin, also brought up the unique challenge of comparability from different versions to the next. So can you comment on sort of lessons learned across these three examples from your perspective?
Yeah, I was actually going to comment on all the positive examples today, because I think we've seen a broad spectrum of different situations where putting the totality of the evidence together in each case led to a decision to grant breakthrough therapy. Rick Pasner was quoted earlier this morning as saying we're looking for things that are transformative. And I think in different ways, each of the designations were transformative, but they may not all equally be transformative. And I think one of the struggles we have in making these decisions is, when is something an incremental advance that you would expect in normal course of developing a new drug to treat a condition, either trying to develop something that's better than what you already have, or develop something where you don't have any treatments at all, versus when is something substantially better and transformative. And I think that can be a judgment call and can be challenging. To me, the hemoglobinopathy, that sounds truly transformative, because potentially those patients could be transfusion-free. You may actually have cured their disease. I don't want to go too far because I don't know all the details, but that seems really like a big deal. But other things are a big deal too. In some of the cancer cases, you're taking people who have no available options and giving them response and benefit. Unfortunately, in many of those cases, you're seeing that the responses don't last very long. So that's a different form of transformative benefit. In some of the cancer cases we're seeing today, particularly some of the immunotherapies, we're seeing the potential for some really long-term responses. So there, often the response rate isn't that impressive, but the tail of the curve for survival has been impressive. The malaria case, on its surface, you're looking at it saying about 90% response rate versus 75, 77% response rate. Is that transformative, or is that just an incremental advance? But then you look at other aspects of, a single dose treatment versus 14 days of treatment in a disease where it's often in third world countries where monitoring 14 days of treatment is very complex. So your success rate is going to be higher in the real world. So I think they illustrate the different aspects of what could be viewed as a substantial improvement, but they're not all the same. They don't have the same flavor. The neurology case I thought was particularly an interesting example, because there they had a significant effect on a biomarker, the thigh volume, and they also had some improvements in clinical measures, and that's in the face of a disease that has no treatment that is relentlessly progressive. So what would be transformative there versus just an incremental improvement, I think, is a more difficult distinction. And we've had a lot of these discussions inside the agency. When you have absolutely no available therapy and you now have something that works, is that by definition of substantial improvement? And how do you make those distinctions? It becomes very challenging. But I think the cases have been very illustrative of the complexity, but also the totality of evidence that you need to look at as you're considering each individual case.
Great, thanks, John. We're going to go ahead and open it up to the audience. If you have any questions about these products, there were a lot of interesting characteristics for those. As you line up, I will turn one more question to John Powers. John, you've patiently listened to all of the presentations throughout the day, and you did mention some issues with respect to antibiotic drug development, but is there any additional thoughts you have that can help manufacturers or researchers in the field of antibiotic drug development better understand how the evidence for and against the breakthrough therapy designation might be important for them to take away?
I was thinking about one thing in that, I thought the example of Tafenoquine was a great one that fit into this paradigm, but the question then comes up, are there other things like the Get Antibiotic Incentives Now or GAIN Act, which was part of FDASIA as well, that maybe guided people down that pathway instead of down this one? And what seems to be a major distinction between breakthrough therapy designation and GAIN is the idea of substantial clinical improvement. Now, some of us lobbied very hard to try to get substantial clinical improvement into GAIN as well. And so the real question is, how are we benefiting patients by going this direction where we're not coming out with things that are better, yet we assume they are better in some other population that weren't studied? I think there's some interesting, I'm bringing this up because I think it would be an interesting comparison to compare drugs approved through GAIN versus those that go through the breakthrough therapy designation pathway. And what do you actually get out of each of those pathways? And one of the things that struck me today that's a big difference is that these studies are actually done for breakthrough therapy designation in the actual patients who are hypothesized to benefit. What we've seen from GAIN is studies done in a population that already has effective therapies, and then maybe we extrapolate to some other population that doesn't. I thought we almost got close to that with Tafenoquine by saying maybe there's the potential for less hemolytic adverse events in people with G6PD deficiency. We're forgetting that this breakthrough designation for this drug was granted in 2013. There's a lot more evidence now. There was a publication just published in Lancet just recently on Tafenoquine, and didn't, to my eye, show any difference between hemolytic events. But that's different. The results are different than getting the designation at the beginning. And I want to make that distinction too. So anyway, that's where I think that it would be nicer to see more people develop infectious diseases that go down this pathway.
Great. Bob?
I have a benzoate question. The thing that the company thought they had shown for the first time ever was an effect on negative symptoms. And if there's a breakthrough aspect of it, surely that's what it is, not the possibly small additional effect on positive symptoms. I'm throwing that out as a hypothesis. It's clearly nice if they were to show add-on benefit in all aspects of it, cognitive function and all those things. That would be wonderful. But suppose the company said, no, the most efficient thing we could possibly do, the fastest way we can get to the end, is to show an improvement in negative symptoms, especially if we enrich the population with people whose negative symptoms didn't respond on their previous therapy. Are we saying that's no good? That wouldn't do? I mean, they've accepted a broader look, and that's fine, and that's virtuous. They'll learn all about it. But for example, a study to focus on improvement in negative symptoms, where negative symptoms is the primary endpoint, is going to be a smaller study than a study that tries to show a benefit on all of them. Not okay? I just think it needs some discussion.
Yeah. May I?
Yes, please.
Yeah, in real life, in fact, it's the negative symptoms is harder to show efficacy than, say, the rest. And cognitive symptom is the worst. So we do have the signal on everything, but cognitive symptom signal is smaller. From our perspective, I think you try to pieces the syndrome is dangerous, dangerous in a way that you may not even ever get a treatment purely for positive, purely for negative, or purely for cognitions. That's I think it's a lot of academic interest in that, but that may not be the.
I'm asking something different. Suppose you were the first antipsychotic drug ever to show a nice effect on negative symptoms. Okay, that would be a big deal because all of the other drugs have failed to show that. Isn't that important? Don't we think that would be an important thing?
Or is that a question
by
for
itself?
me, or is it a question for,
I'm
for you?
asking you, Lucas, and and John for that
Could I ask
matter.
a corollary of that? Because this comes up all the time, Bob, and that is that I was thinking about this earlier in a previous discussion. When you start narrowing the indication that focuses on a piece of disease and not the whole thing, don't you also want to know that you didn't make the other piece worse? And so I guess that's the question would be, if you would focus on just negative symptoms, doesn't have to say you have to show a positive
Okay,
effect.
that's
on
not the
positive
same as showing a benefit in that group.
Right, exactly. That's
a
what
good
I'm saying, though.
question. Good
And the
question.
reason I bring this up is this comes up in PROs all the time, right? So I'm going to develop a PRO for pneumonia. I'm going to focus on cough, but I'm going to ignore shortness of breath. Well, if you decrease cough, but you made the guy's shortness of breath worse, that's not beneficial to people. And so I'm bringing the issue up here of, if you showed an effect on negative symptoms, wouldn't you still have to look at the positive, which is what I think you're saying.
No, but there's a difference between looking at them, seeing that they're not going the wrong way,
Yes.
and knowing that you have to win on that.
Exactly.
And they might, this all goes to efficiency of design and study size. If there's some sense of urgency, you want a study that shows it rapidly, that may mean that what your primary endpoint is could determine how likely you are to win.
It's a very important question. That's why in the past 10 years, there are 3 consensus meetings. Academia, NIMH, EMA, FDA. So it's no brainer to us that when we have the data, we go for negative symptom in a way. Because, again, I'm repeating this, is that in our daily life, when we practice psychiatry, we do add-on treatment all the time. The novelty is not that great when you do add-on in real life. But the novelty of a negative symptom is a big deal. That's I agree. But for us, I think at the end, it's what matters to the patients. This treatment, good, it's good for negative symptom, also good for the rest, then that's great. As a practitioner psychiatrist, I have that as priority.
I also presume, Lucas, that if they did study all of them and were the first drug ever to have a favorable effect on negative symptoms, they'd be allowed to say that in the label. They wouldn't just have to say we work on everything, right?
Yeah, well, that's part of how we were thinking about it. How are you going to label this drug, right? Why would you just label it for negative symptoms?
I didn't say just label it for negative symptoms.
Exactly what
that's not what I asked you. I asked you, would they, if they were the first drug ever to show an effect on negative symptoms, be able to say we work on negative symptoms as well as positive symptoms?
Well, if they show that they work on positive and negative symptoms, that'll be in the label.
Okay. And if they only worked on negative symptoms, they could be labeled for that too.
Right. And that's what that paper describes, Lochran and Levin paper.
But even if they worked in both positive and negative symptoms, they could still be labeled for negative symptoms, as well as positive symptoms, right?
Right. You
I think
described
we're getting a little
what
off
they
topic
want.
here.
If they want on cognition, they get cognition. So we want to make this as easy as possible for clinicians to use and understand what they're going to get when they prescribe this to patients. So.
John Jenkins?
I think the case Bob is describing is another illustration of what I was trying to get to earlier, of trying to make the distinction in a situation where nothing previously has worked for a disease or a symptom. Is any effect in that disease or symptom a breakthrough, or is that an incremental advance that you would expect as part of normal drug development? We've had endless debates, philosophical debates inside the agency about, what is the bar? That's why I gave the definitions at the beginning this morning of substantial and significant. And that may be a good food for thought for the final panel to bounce around some. If you've got a serious disease that previous therapies or there are no previous therapies have had an impact, and then you have a new drug that has any level of significant impact that would be approvable, is that a breakthrough? Or is that just the incremental expectations of the normal process of drug development? And I bring this up because it gets to where you draw the bar, And do you have the 2 or 3 breakthroughs a year that Ellen described when the law was being proposed, or do you have the 80 that we have now, almost 3 years into the program? Because obviously you can't sustain a program where everything is a breakthrough, because you can't have all hands on deck for every program because everything's a breakthrough. So it's a philosophical discussion. We've tried to wrestle with that and come to some sense within, you know, the Medical Policy Council and CEDAR about how we make decisions on these, but it's a challenging one, and some of it depends upon, you know, how you're looking at each case. So it may be something the last panel could grapple with as you're trying to do lessons learned from today's discussions.
Great, thank you. So that's a good point. So the last panel, Mark, you're in the audience, is the big question is, have we brought clarity to what is substantial versus, or transformational, which I think we've agreed might be the same thing, or significant. I'd like to thank all of the panelists. These were great. These 3 cases were just great examples and brought up a range of challenging issues that really do help us understand beyond oncology how can other products really fare within breakthrough designation and what the evidence is needed for that designation. We are going to take a 15-minute break, so come back at 3:45 for our final panel that will summarize the day and take it to the higher level in terms of what companies and researchers really should be thinking about substantial evidence for these breakthrough designations. Thank you.